So now let's talk about the direct microscopic count. Direct microscopic count is a quantitative test and it is helpful in assessing the actual number of bacteria present in the milk. It is a platform test where the microbiological quality of the milk is received at the raw milk receiving dock, and it is a very rapid method. It helps in the estimation of the total bacterial count of the population of the sample of a milk, and it is also used in tracing the sources of contamination of milk. So what does it mean? Directly you take a microscopic count of the milk. Let's see how is it. It's based upon a principle that the examination of the stained thin film of measured volume of milk is spread over a specified area on a glass slide, and the method is useful for the rapid estimation of total bacterial population, including the live as well as the dead cells of a sample of milk. And in this test, the milk smear is prepared in 1 square centimeter area, and it is stained with a special stain called the Newman stain and examined under the microscope. And each microscopic field examined represents a quantitative aliquot of the milk sample. The number of microscopic fields occurring in 1 square centimeter area of the milk smear will vary as the diameter of the microscopic field varies with different microscopes, and this necessitates the calculation of microscopic factor for each microscope separately. So what you do basically is you take you have a glass slide. Onto the glass slide you take a sample of milk, okay, and you spread it, okay, and after spreading it at a particular area of about 1 square centimeter, okay. If you directly look into the milk, you cannot identify anything, but you will have to stain it first. After spreading it, you have to stain it, and the stain that which we use here is a Newman stain. And after that, you at a particular area of about 1 square centimeter area, okay, we will analyze it through the microscope and count the number of microbes which have been present in it. Now, since the staining is also being done, the microbes will be colored, so you will be able to identify it. Now, okay, of course you cannot just simply prepare a smear. You will need to, even the live as well as the dead cells can be viewed in this case, because the dead cells are not going from there, okay. And when it gets stained, what happens? Both live and dead cells will get stained by the Newman stain, okay, and we will be able to identify it. And of course also the site of contamination can also be analyzed, because even in that site also, if you find the same microbe, you can say that okay, the chance of contamination is from that particular site. For the preparation of a slide, now you take 0.01 ml of milk with the help of a sterile Breed's pipette and spread it evenly on a grease-free slide of 1 centimeter square marked area on the Breed's line, and dry the smear on a warm surface for about 40 to 45 degrees centigrade. The symbol has been wrong over there, and do not heat fix the slide on direct flame. Rapid drying results in cracked surfaces on the film or the film peels off during the processing. So you just keep the slide as such. You do not give heat fixation, but of course you need to fix it out, okay. So you just keep it over there, or dry the smear on a warm surface for about if it's a warm surface. Like if you don't fix it, what will happen? Like heat fixation means it will result in the formation of, in the direct flame, if you heat fix it, what happens? The protein and all that will peel off from it. So you cannot say that you cannot be without fixing. You have to fix it, because when you add a stain, what happens? The microbe also will run out. So what you do here, keep it on a warm surface at 40 to 45 degrees centigrade for some time, so that it becomes dry. After that, you immerse the slide in Newman stain for about 1 minute. Now the Newman stain will remove the milk fat, fixes the smear, and stain the bacteria in a single operation. And the tetrachloroethane of the stain will help us to dissolve the milk fat globules. Ethyl alcohol fixes the smear, and methylene blue stains the smear. So we have 3 different ingredients in this particular smear, and simultaneously when you put this stain, sorry, not the smear, the stain. In the stain there are 3 different components which will help us to analyze it. Now for example, we have tetrachloroethane in this particular stain called the Newman stain, and that will dissolve the what? The milk fat globules. And then we have ethyl alcohol which will fix the smear. What is fixing the smear? Okay, you know what is it. And methylene blue will stain the smear. So these 3 things can be done by the same stain called the Newman stain. And after that you count different fields. Through the microscope we have to manually count the amount of microbes which have been present. So if you have a clumps less than 0.5, the fields to be counted is 50, and 0.5 to 1 clump is there in a field, then you have to be count 25, you see, and 10 to 30 clumps are there in a field. What 5 fields have to be counted. The field means the area, you have to look through the microscope, and that is referred to as the field. Okay. And the grading of the milk based upon the direct microscopic count as per the BIS standards is that if you have a count less than 5 lakh, okay, clump counts, okay, if the count has been found to be less than 5 lakhs, okay, it is saying that the quality of the milk is good. And if it is 5 lakhs to 40 lakh clumps are there, it is fair, okay like. But 40 lakhs to more than that, almost to 2 crores, right, that is a poor quality, and beyond that it is a very poor quality. So you take the milk here and you just, what you did, you prepare a smear using a Newman stain, and you take a count and you look the number of clumps in it. Depending upon the clumps you grade the quality of the milk. That is a direct microbial count. Microscopic count, sorry, direct microscopic count. What are the advantages here? DMC is widely used to screen incoming raw milk at as a platform test to determine whether the milk has an acceptable or a legal bacterial load as per the BIS standards, and it is easy to perform and less time is required to perform the test. Large number of samples can be screened in a period of time, and it is useful in providing the estimated counts, types of bacteria and the somatic cells in the milk. And it is used as a guide in identifying the types of bacteria present in milk sample. And you will be able to find out the possible source of contamination of milk also. I told you, you can take the smear from one area as well as from the other area also. So depending upon the source, from if one area has the clumping and the microbial count, you can say that that is the site of source of contamination can be guessed out. And a record of the test in the form of a slide for legal verification can also be maintained. The equipment and manpower needed to assess a microbiological test is been found to be less, and possible it is able also to find out mastitis and the presence of other somatic cells and all in the milk samples. That is, if there is inflammation of the mammary glands, okay, that will result in the condition called mastitis or the inflammation of the mammary glands, that's called mastitis. In that case also there will be a presence of somatic cells or the body cells in the milk. They can also be detected by the direct microscopic count test. The disadvantages, it is not considered as a foolproof or a legal method, and it is of course a strain on the eye of the operator, because every sample when you look you have to have you get exposed and the eyes become weary. And the test is not reliable as both viable and non-viable cells are counted. You know what is the difference between a viable and a non-viable cell? A viable cell is a one which is living, and a non-viable cell is a killed cell. And this method is not suitable for pasteurized milk also, and experienced person can carry out only this test, because if the person is having no proper eyesight, what happens? You won't be able to analyze and tell it. And the results are not reproducible because the microbes are unevenly distributed in the smear, because depending upon the smear pattern or how you prepare the smear also, the success rate of this direct microscopic count will vary. So it is not a reproducible one. The results are not reproducible. Any experiment is said to be reproducible is if you get even on repeated trials you get the same result. That is something what is known as reproducible. So now if I prepare a smear in one way and another person prepares in another way, a smear of in another way of the same milk, might be we will get different results. In such a case, what happens? It is considered that this method is not reproducible. Okay, and moreover, the microbes will get unevenly distributed in the smear. So, depending upon, and such a thing will result in different types of clumping. I think it's clear right now. We will go to the methylene blue reductase test in the next video. Thank you.